Today we are looking at medical instruments. It could be complex but yet very essential. Well, what if it could be made easier? Welcome back to MedSense, where we make medicine make sense. And today we are going to be dissecting the concept of medical instruments. A microscope is an instrument used to magnify small objects, allowing detailed visualization of structures not visible to the naked eyes. Now note that in the course of this video, I'm going to be pointing out some likely exam questions for those that intend to use this video for the preparation of their exams. So, types of microscope. This could be an exam question, could present in this form. Types of microscope. We have the light microscope, electron microscope, fluorescence microscope, confocal microscope. But we're not going to be going into the details of each and every one of them. This is another question that usually presents parts of a microscope. Looking at the diagram, an eyepiece, nosepiece, objective lens, slide holder, it always repeats in any exam that has to do with instrumentation and a microscope comes out. You must see parts of a microscope, so take note of the parts. The next on the list is a syringe. A syringe is a medical tool used to inject or withdraw fluids from the body. Now, the major question that comes out on syringe, if it ever presents, is not going to be types. It's going to be parts of a syringe. They can draw a syringe and tell you to label the parts A, B, or whatever as the case may be. But take time to know the parts of a syringe. Most importantly, how to use it as well. An electronic weighing balance is a device used to measure weight or mass accurately. Emphasis on accurately. It works using an electronic sensor instead of the traditional mechanical weights. Types of electronic weighing balance. We have the precision balance, the analytical balance, and the micro balance. The most common amongst all of them is the analytical balance, and we're going to be seeing the diagram shortly. So this is the analytical balance, and looking at it, this is the diagram. So it could also present and a part to be labeled, and they will tell you identify the instrument, identify the part A. So it has a glass door handle, power buttons, draft shield, balance pan, which is more or less the most important part of it. The principles of action of any instrument is very important. Now, for this weighing balance, the principle of action is when you place an object on the weighing pan, which we saw in the diagram, the load cell senses the weight and sends electrical signal to the system. The system converts this signal into a weight reading and displays it on the screen. That is the principle of action for this electronic weighing balance. A pH meter is a device used to measure the acidity or alkalinity of a solution. It gives a precise pH value. Next, parts of a pH meter, we have the electrode, the display screen, calibration knobs, power source, and over here is the diagram or a pictorial representation of what a pH meter looks like. Principles of action of a pH meter. The electrode contains a special glass membrane that reacts with hydrogen ions in the solution. This reaction creates a tiny electrical voltage. The pH meter converts the voltage into a pH value and displays it on the screen. Next, we are looking at a stethoscope. A stethoscope is a fundamental medical instrument used to listen to internal body sounds, such as a heartbeat, lung sounds, and blood flow. This is the parts of a stethoscope. Take note of it. It will always present in an instrument. A spectrophotometer is an analytical instrument used to measure how much light a substance absorbs at different wavelengths. Principles of action. It works majorly on the Beer-Lambert's law. That is the principle of action of the spectrophotometer. Now, for the concept of its function, when light passes through a sample, some wavelengths are absorbed while others pass through. The spectrophotometer measures this absorption to determine the concentration of the sample. Now on the next slide, this is the pictorial representation of a spectrophotometer. The parts of a spectrophotometer, we have the light source, we have the monochromator, we have slits and lenses, sample holders, and photo dictator. Now we're looking at how a spectrophotometer works. The light source emits a broad range of wavelengths. The monochromator selects a specific wavelength. The light passes through the sample cuvette. The detector measures the intensity of light passing through. The processor calculates the absorbance. The results are displayed on the screen. Advantages of a spectrophotometer over the colorimeter. This is a question that will always come, and they can tell you compare and contrast. Now, the advantages is that the spectrophotometer will give a high accuracy result, can analyze both colorless and colored solution, wide range of application. It has a non-destructive testing method. For the disadvantages, number one, it's a very expensive equipment. It requires regular calibration and maintenance. When the samples need special coverts, it doesn't use the regular. On the other hand, the colorimeter is an instrument used to measure the absorbance of a specific wavelength of light by a solution. A colorimeter works on the principle of absorption and transmission of light based on the Lambert's law. The concept, a light beam of specific color, emphasis on specific color, passes through a sample. The solution absorbs part of the light and the remaining light reaches the detector. The absorbance is measured and correlated to the substance concentration. Now, this is the picture of a colorimeter. Please familiarize with these both pictures so that you don't mix them up. Parts of a colorimeter, we have the light source, filter, sample holder, detector, display unit. Now, another thing worthy of note is that a colorimeter uses a filter, but a spectrophotometer doesn't use a filter. How a colorimeter works: the light source emits a beam of visible light. The filter selects a specific wavelength that matches the color of the solution. The light passes through the sample in the cuvette. Detector measures the intensity of transmitted light. The processor calculates the absorbance and displays it on the as the result. Advantages: samples are simple and easy to use, affordable and portable, quick and reliable, requires minimal sample preparation. Just the direct opposite of a spectrophotometer. Disadvantages: limited to visible light, less precise than a spectrophotometer, only works for colored solutions. Now, on this slide is where I gave a detailed comparison between the both of them, a spectrophotometer and a colorimeter. So zoom in to see for the light source, the comparison between the both of them, principle, monochromator, accuracy, application, and the rest of them. The next on the list is a tissue embedding center. A tissue embedding center is a specialized laboratory workstation because of how big this machine actually is, used to embed processed tissue samples in a solid medium, usually paraffin wax, for sectioning and microscopic examinations. Reasons for tissue embedding. Really, why should tissues be embedded? Now let's find out. For structural support to fragile tissues, proper orientation for accurate histological sectioning, preservation of tissue morphology for microscopic analysis. Now on this slide, this is the picture of a tissue embedding center. This picture doesn't exactly do justice to how big this machine actually is in reality. Now, parts of the tissue embedding center may or may not be necessary, but still look into them. The next one, steps in tissue embedding. Now, this may or may not also be necessary, but I thought it to include it in this video so that you would have all the knowledge at your disposal. It's better to be over-prepared than to be under-prepared. A cryostat. A cryostat is a specialized laboratory instrument used to cut extremely thin tissue sections at low temperatures. Now, emphasis on extremely thin tissue sections. Now, this is the picture of a cryostat. It's necessary you know what the machine looks like as well. Principles of a cryostat. A cryostat functions as a microtome inside a refrigerated chamber. Now we are also going to look at the microtome. Now, maintaining temperatures between -20 to -30. If you could remember earlier, we said it functions at very low temperatures. This preserves tissue morphology and prevents structural damage during sectioning. Parts of a cryostat: we have the refrigerated chamber, the microtome, specimen holder, anti-roll plate, and the rest of them. An autoclave is a specialized device used for sterilization by applying high-pressure steam to kill bacteria, viruses, fungi, and spores. Principles of an autoclave works on the principle of moist heat sterilization using high-pressure steam to destroy microorganisms. Now, very important, the standard conditions for an autoclave to function is from 121 degrees Celsius to 134 degrees Celsius. The pressure is 15 to 30 pounds per square inch, and for 15 to 30 minutes. Very necessary. These standard conditions most likely presents in exams. Now, this is the picture of an autoclave as well. So that's all you need to know. Uses of an autoclave for sterilization of surgical instruments. Decontamination. We also have sterilization of culture media, sterilization of pharmaceutical products, etc. For the water bath, a water bath is a laboratory equipment used to heat samples at a constant temperature using heated water. So this tries as much as possible to maintain a constant temperature. Now, this is the working principle, works on the principle of indirect heating. A container filled with water is heated to set a temperature, then the sample is placed in a test tube inside the water bath. The heat is transferred indirectly, don't forget, gently, evenly, to prevent overheating or burning. So this is the picture of a water bath. Types of water bath: we have circulating water bath, non-circulating water bath, shaking water bath. Uses of water bath: we have incubating samples, melting, warming reagents, maintaining reaction temperatures. Now let's finally talk about the rotary microtome. We've made reference to it at some point in this video when we discussed the cryostat. Now, it's a laboratory instrument used to cut extremely thin sections of embedded tissue samples from microscopic examinations. In what machine did we talk about the embedding of tissue samples? Now, this is the picture and it's labeled, as a matter of fact, of a rotary microtome. Now, uses in the histopathology lab, which is the major place it is used for preparation of tissue sections for microscopic diagnosis, research laboratories, forensic investigations, and all that. Now, that's for a microtome. Now we are going to be looking at centrifuge next. A centrifuge is a laboratory device used to separate components of mixtures based on their density by applying centrifugal force. So they will always make reference to this centrifugal. Questions will always revolve around it. Maybe in objective questions, you will be seeing things like, is it by applying centripetal force or centrifugal force? Don't forget centrifugal force. Principles of centrifuge works on the principle of centrifugal force. Again, it has been reiterated. The sample is spun at high speeds of 3000 rpm. Now, forcing denser particles to move outward and lighter components to remain at the top. The separation occurs based on density differences in the samples. Principles of a centrifuge done. Now, this is the picture of a centrifuge, and then uses blood separation, which is majorly where it is used. It can also be used during a microbiology testing, DNA and RNA extraction. So that is all for our medical instrumentation. Thank you for watching up till this point. Do not forget to like and subscribe to our channel. Stay tuned, on the notification bell as well, so that you will be informed when next we post videos. Thank you very much.